All right. So this is very exciting. This is a real hybrid talk. I think one of the very few we've had so far. So I am Mario Gemete. I'm an assistant professor in the Institute for the Ocean and Fisheries and in the Department of Statistics. And so without further ado, I'm really excited to present Dr. Vianey Barajas today. Dr. Barajas received her PhD in statistics in Iowa State University. She is now an assistant professor at the University of Toronto. She's cross-appointed in between the Department of Statistical Science and the School of the Environment at the University of Toronto. She's also a faculty affiliate of the Vector Institute and a faculty member of the Center for Global Change Science, also at the University of Toronto. Vianey I'm very excited that she's presented today. She's a person that I follow the work very closely. She's always at the leading edge of development of animal movement models. She works a lot on marine species, and I'm always excited when a paper came out. And recently we've become colleagues, and we're now working on a big project together. And so I am very excited for all of you to have a chance to hear about her wonderful work. So Vianey, go ahead, take the show.
Okay, thank you. I'm not sure if the mics are working, if everything's okay. I hope it is. Thank you so much to Marie for the invitation. Thank you so much to everyone also for all your help and being here in person today. I realize that this is actually not common, but I was actually very excited to get on the talk because so hidden Markov models. I'm talking today about hidden Markov models. Mathematically, formally, what are we actually talking about? It's a double stochastic process composed of an observation process, which is the data that we collect from the animals, so their positions, their acceleration, etc. And there's an underlying latent state process, which we take to be in ecology, biology, the animal behavior. So when they're performing different behaviors, that manifests itself in the movements that we see in the data. The observations can be discrete, continuous, measured with error, multivariate, and so on. But the general idea is that you have some data streams collected over time, and they're a product of the animal performing different behaviors. As I said, CT is a discrete random variable, so we're looking at a finite set of behaviors that we're actually interested in. So the graphical representation that you'll see in papers is typically this structure. We love putting this graphic in our papers. There's an observation y sub t and a state process z sub t. So the idea is that there's actually persistence in an animal's behavior, depending on the temporal resolution. So for example, if they've started to eat, they'll likely remain eating for a long time. If I work with some sheep data, when they start eating grass or start eating other plants, they're going to eat for there's going to be persistence in that behavior. We want to make sure that we take that into account. And the observation process in the most basic form is taken to be conditionally independent of each other given the state. So what is it? The essence of a discrete time finite state hidden Markov model is you have the number of states, which we have the state defined distributions, transition quality matrix, and initial state distribution. So they're kind of like the key parts of what an HMM actually is mathematically. On the right-hand side, oh, I didn't do that. Awesome fun.
I'll fix it. I think someone is annotating your slide.
They don't like that. They don't like the general interpretation. Okay. It's okay. You can tell me this doesn't make sense. Here's getting bad feedback. So for me, the general interpretation is that the number of states generally refers to the number of movement patterns that we're actually interested in capturing in our data. We have the probability distribution for realization from the different patterns. So when an animal is traveling, there's a certain distribution of speeds that correspond to traveling behavior. So what does that actually look like? We have the state switching dynamics across the pattern. So how does an animal go from one behavior to the next? What is that sequential process? And the initial state distribution, what is happening at the first observation? So when we first observe the animal, what is it doing? That's what we're trying to infer. So one of the great things about HMMs is that it's kind of a way to do clustering with temporal dependence. Because without telling the model which observations correspond to which states, it can actually infer that for you. Right? So you have your time series, it can be positions, acceleration, and so on. And there's different algorithms associated with connecting the observations, to the different set of states that you're interested in. And that's called state decoding. So what does data actually look like? So what is a hidden Markov model? It's easy to talk about the mathematics, but sometimes a lot more intuitive when you actually look at data generated from the process. So let's say we're simulating from a 3-state hidden Markov model. We have our state-dependent distributions or normals. So there's 3 different normal distributions that are generating our data. We have our initial state distribution. It's 1/3, 1/3, 1/3. So it's equally likely that the animal is starting in one of the 3 behaviors. And we have our transition probability matrix that tells us the amount of time that the animal will stay in that behavior before switching. So if you have a time series and you say, well, let me remove the temporal component, just plot a histogram. What the histogram will likely look like is it'll have these multiple modes. There's a possibility, right? So you have your gold state, you have your blue state, and you have your green state. And you have data generated from those 3 distributions. If you look at the first 500 observations, and 2,000 observations we generated, what we'll see is that we have gold points clustered, green points clustered over time, and blue points. So the idea is that you're not just simulating data randomly from those 3 different components, but when you first have a gold observation, you're likely to have a few of those gold observations over time. Then you switch. Then you're likely to have a few green observations. Then you switch. Then you're likely to have a few blue observations. So for me right now, as I'm making this talk, I'll likely start pacing. I won't pace once, I'll just kind of pace for a little bit. And then I'll be like, okay, I need to explain this, so I'll stop. So I'll stop for a while. I'll move my hands for a while, etc. So one of the big papers that I reference is a paper by Juan Morales, Ecology 2004. So extracting more out of relocation data, building movement models as mixtures of random walks. So hidden Markov models are often referred to in different contexts. So a mixture of random walk is actually a hidden Markov model in this context. So there's movement data from 4 up in East Central Ontario that were reintroduced from Alberta. They had daily positions, which they converted to step lengths, which is the Euclidean distance between 2 points, and turning angles. And they tried to infer 2 states, something referred to as encamped and other exploratory. So the idea is that encamped would be, or how we would see it in the movement data, is high turning angles and short step lengths. That would be one movement pattern that we want to capture in the data. We also have exploratory behavior, which is actually between 2 points. They're going to travel multiple kilometers and have a turning angle close to 0, indicating that it's really directed motion. So there's 2 behaviors that we really wanted to capture and have exploratory. And the way that they translated that into the data is these turning angles and step lengths. So the general ecological insights gained is that, well, they can connect the observations to different patterns in general and HMM via state decoding. They can estimate the probability mass or distribution functions for the different patterns. So for encamped behavior, we actually have a distribution of step lengths that corresponds to that behavior. They can model persistence in a behavior, the state switching behavior, and the amount of time in that state before switching. So how much time will they spend in an encamped behavior before they move to an exploratory behavior? And in a very, 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 very loose sense, you can estimate the number of patterns seen in your data. For the ELK, they also incorporated the probability of encamped exploratory states. They allow that to vary by habitat. So depending on the habitat, they would say, well, actually, this affects the behaviors that the ELK would exhibit. So you can incorporate covariates to understand drivers of these patterns. So hidden Markov models today and hidden Markov models for sharks. So since I'm here with you all, I'm going to present a lot of the shark work that I've done. But generally, hidden Markov models in statistical ecology have gone a long way from just this sort of two-state hidden Markov model for in CAM's exploratory. There's hidden Markov models are also commonly referred to as Markov switching models, state switching, regime switching. There's one of my PhD students is working with multivariate outcomes, modeling via copulus. You can do hidden Markov models in continuous time. You can have flexible state-dependent distributions, which I'll show in a second. These multi-scale HMMs and so on. So my first paper, and I bring this up not because I want to brag on my first paper, but because I was a PhD in statistics, PhD student in statistics, who was obsessed with sharks. And I still have a slight shark obsession based off my math, my tattoo, so on. But I always wanted to work with shark data for some reason, right? I was like, this is the coolest data. I don't just want to develop statistical models, but I want to work with marine data. So I was able to connect with a few people and was able to work on this white shark project. There were 14 individuals who were collecting positions. So they were tracking the shark in South Africa, 5 males, 9 females, multiple sizes, and they will track over multiple days and track one at a time. So the basic idea was to fit a two-state HMM to capture area restricted search and traveling behaviors. As you can see, these are the state-dependent distributions, the densities, and the turning angle densities for the two different states. So we kind of have the same as sort of the elk of we have one distribution that has longer step lengths, one that has shorter. The turning angles have slightly different. There's one that's centered around 0, and other one that corresponds to a much higher turning angle. Now, sort of an advancement from the basic two-state HMM of the elk is that we use mixed effect hidden Markov models. So the key question was, what is the effect of chumming on the white shark movements? So we put it in the model, the so-called discrete valued random effects. And covariates, so sex, chum, time of day, and the length of the shark. So the intuition behind the discrete valued random effects is that you want to account for variation across the tracks. But in particular, for short tracks, sharks may exhibit very different movement strategies across different days. And that might just be natural variation, depending on if they've eaten recently, depending on multiple factors that we might not be able to measure. But we might want to take into account that heterogeneity before we make any inference about any covariate effect. So the final model was a sort of two-state HMM, just like the elk. We had actually 3 random effects groups, which I'll describe in a second, and with a few covariates included. So the outcome, so the 3 random effects groups is actually in that purple box, epsilon sub ik. Effectively meant that each track was generated from one of 3 hidden Markov models, where the state-dependent distributions were the same, but the transition polity matrix differed. That just meant there was slightly different state switching behavior across the 2 different states that we captured. And then after taking that into account, we incorporated these effects in the movement dynamics. And one of the key things is that we have these 3 different transition polity matrices. So on the right-hand side, these are the random effects groups. So the RE1, RE2, RE3. And we actually saw that the female sharks tended to exhibit more of this first random effects group. And there were some slight differences, which the biologists took to mean that they had different hunting strategies or just different types of movements that they exhibited. We have just this points of the males and the females. And here you sort of have a box where we have one trajectory of the female shark. So you see there's this gray pattern. That's a sort of traveling behavior. So the HOM tells us that part of the time series is traveling, which is directed motion. And then everything else where it kind of had more area restricted search movement is all in black, as told by the HMM. And the red dots here is chumming. There's chumming activity. And there's the little shark fins that are actually in the paper indicate a predation attempt that was observed by the boat. So it's the same two-stage hydromorphic model as the ELK, but now we've included this discrete value random effects. I've also worked on oceanic WHITETIP tracks, in particular the accelerometer data. And we started simple. We summarized accelerometer data and calculated ODBA, so overall dynamic body acceleration. And the biologist asked, well, can we model this with a hydromorphic model? Accelerometer data, as you all know, is very, very fine scale. There's a lot of details and a lot of data, which means that when I try to fit a hydromorphic model and I try to select a state-dependent distribution, something like a normal or a gamma, it's actually not going to work very well. So what I turn to is these semi-parametric distributions. So a 3-state HMM will now have distributions that look slightly different than what we usually work with. But it provides a lot more flexibility in the types of patterns that we can actually capture. And we can go back to the time series and we can do our state decoding, where we have an observation that corresponds to the green state, the blue state, or the gold state. Now, this is a hard decoding, meaning I'm saying this observation can only come from the state. We can also extract probabilities. And the idea behind the ODBA project as well is that we eventually overlaid these decoding onto the depth to see higher values of ODBA. What were the sharks doing? Were they diving? Were they swimming along, etc.? This is where I learned the term negatively buoyant for sharks, which of course I'm coming from a math and stats background and I didn't know. I've also used these semi-parametric HMMs for gray and BLACKTIP reef sharks. So above are these 4 distributions for the BLACKTIP sharks, below the gray reef sharks. Also for overall dynamic body acceleration. So really overall dynamic body acceleration with this fine scale acceleration data really turned me on to semi-parametric HMMs because I failed for months trying to fit parametric hydromorphic models. And what we wanted to do here actually was we did something very simple. And sometimes we don't need to do the most complicated thing. We try to say, well, there's peaks at ODBA and there's periods of ODBA that are quite low. So what we're trying to do is just capture relatively higher ODBA values, more active, and relatively lower ODBA values, less active. And try to say, well, then how does that vary across time of day? So if we say state 1 is lower activity and state 2 is higher activity, we can incorporate covariates like time of day to understand when the sharks are exhibiting higher activity and when they're exhibiting lower activity. And I believe that these 2 shark species are inhabiting the same area. So we wanted to compare also, are the gray reef sharks and the BLACKTIP sharks, are they active at the same times of day or is it different? And you can also see on the x-axis that the state 2 for the gray reef sharks, I think this is the gray reef sharks, varies quite a bit more in probability of state 2. The activity goes from 0.2 to 0.4 here, whereas there's not as much change for the other species. These are very low sample sizes. So I'm not saying that in general, all sharks are going to do this, but for the data that we had, this is what we were seeing. And what we also then did is we have our ODBA decoded states. And what we wanted to do then is say, well, we have depth and temperature. So let's just make a scatter plot, which I made into these grids. And say, well, for different values of depth and temperature, where are the higher activity ODBA values as described by state 2, and which ones are on state 1? So the percentage of the proportion of time spent in state 2 is in red. So you can just see what combinations of depth and temperature, they tended to exhibit more state 2 than state 1. So again, this could be slightly descriptive, slightly exploratory. And for me, that's okay. It's just useful information. I've also been working on something called multi-scale HMMs, who are now referring to as particle Markov models. And the idea is just to extend it. Right, so we're talking about extensions today. We have our observation process, just like before. We have our fine scale state process, which before we just called the state process. Now I'll call it the production state process. And the idea now behind this extra layer at the bottom is that there's a sequence of states that produces a sequence of observations that actually all correspond to the sort of larger scale behavior that I'm trying to capture. So for instance, for me, in my mind, I would like to see this movement data if I knew when a shark was actively hunting. But the data was collected, like accelerometer data, maybe we have a data point every 3 seconds. I assume that maybe hunting for certain types of data sets, it's not just going to be high peaks, like high odba. It's not just high odba for a long time. Maybe it's interspersed with some lower values of odba as well. But maybe there's like a whole sequence that I'm trying to capture. And that whole period really corresponds to what I'm trying to capture in the movement data. So I was like, well, this extension is actually quite easy. And part of the actual motivation, though, is for these baby snakes. So you have this neonate garter snakes, which my friend, this is my friend's PhD, put a red dot on the snake. This is experimental design. And you'll see a Q inserted with a different smell. And what he actually wanted to measure is, he has 2 different time series, before the Q and after the Q for movement. And he wanted to really compare, not at the fine scale level, what if their behaviors changed, but he wanted to know, is this time series behaviorally different from this other time series? So we represent each time series as coming from one of however many possible hidden Markov models. And then we kind of can do some clustering. Are they actually the same hidden Markov model or different? The 2 different time series. We generally saw that the baby snakes actually slowed down after the Q. And it could be that part of it is that they go into this arena, which is kind of like little buckets, and they get worn out. They're moving around a lot, and they've adapted and they kind of slow down the second time period. Some snakes were slow from the beginning, so they didn't actually change hidden Markov models. But again, these are our multi-scale HMMs. Are they changing their movements across time series? Are they consistent in their behavior at a coarser time scale? This is the idea that we were trying to get at, and part of the motivation for these multi-scale HMMs. But what I love about hidden Markov models is that there's coupled HMMs, factorial HMMs, all sorts of HMMs that can all be written as just a basic hidden Markov model. Okay? It's just presented slightly differently. And I think one of the easiest ways to maybe see this, which is probably not very easy, but is in the transition probability matrix of this multi-scale hidden Markov model. So on the diagonals, these are actually matrices of different hidden Markov models. So actually, a multi-scale HMM is a way to switch between hidden Markov models over time. And then each hidden Markov model has n production states, which really could be like a 2-state hidden Markov model in camp exploratory. Maybe there's different days where an animal will perform different, will have a different state switching dynamics across and camp and exploratory behavior. And you can model that over time. And also, part of the multi-scale HMM, which I won't present today, is that different covariates, the same covariate, can have a different effect on the animal depending on something that we don't measure. So maybe the presence of a seal for a white shark that has just eaten will have a different effect with the presence of a seal for a white shark that's hunting. And we can try to account for that possible different effect with these multi-scale HMMs. This also led to multi-scale HMMs for time series collected at multiple temporal resolutions. And Adam is also working on this. He's a postdoc at St. Andrews, and he wants to call them hierarchical hidden Markov models. So I really apologize because I'm calling them multi-scale HMMs, he's calling them hierarchical HMMs. We're making things worse for everybody else. But the idea is now we want to combine movement data collected at different temporal resolutions. So if you have accelerometer data, how do you combine that with GPS data or depth data, where one is collected at a much coarser time scale than the other? So now we're going a little bit nuts with the graphical structures. We're extending, we're including. So part of that motivation, again, back to the sharks, was working on horn sharks. So this is a study by Emily Neese, who is now a PhD student, and she was at California State University Long Beach at the Shark Lab with Chris Lowe. What we wanted to do is these three joint states. We had accelerometer data at a very fine temporal resolution, and they were tracking the horn sharks and collecting a position about every 2 seconds. And the idea was that, well, you can actually see when the horn shark was sort of moving positions or not. But if you only had the GPS data, you might think, well, it's not moving, so it must be resting. That's what people do in terrestrial animals. An HMM where it's like not moving very much, it must be resting. Well, the horn sharks, of course, they can stop and they can just, they don't have to move positions, they're eating something. So we have these two different states, these joint states on the right, where there's no change in position. And one is where the horn shark is stopped. And the other is actively doing something. And together we can actually build those behavioral states. So the next, then a bit of a depression from the sharks, the sheep. We're wanting to incorporate feedback mechanisms in the study of animal movement. As you all know, there's this sort of physiological component and probably social component of an animal in the decisions it makes for its movement patterns. So for the merino sheep, we have movement data, GPS and accelerometer data. We want to incorporate the body fat percentage as a proxy for body condition. So back to hidden Markov models, of course, always hidden Markov models. But here we have our observed movement, which maybe we could take to be GPS data. Our unobserved behavioral state, which tends to be for terrestrial animals very simple, not short step lengths, high turn angles, or just very short step lengths that tend to be resting or zero. We have this area restricted to search, or we have traveling. We generally try to just capture 3 states. What we want to know is, for the sheep, for different body conditions, how do they actually perform these different behaviors? Does it vary over time? So they have a flock of sheep in Argentina. They have some sheep, they started some sheep in poor conditions and some in better conditions. And they wanted to understand how they actually move. For me, of course, back to the sharks, love to do this for sharks. And I know there's a lot of like shark thermoregulations that should have an impact on how it moves. That people might not account for now. You might just think, well, time of day, habitat, but there could be other processes, right? So for the sheep, what we're doing is we're including body fat percentage. And so this is for my PhD, I had to learn about sheep dynamics. But energy levels are going to increase according to foraging intake, decrease locomotion costs and daily maintenance. We construct this energy balance, and we're actually trying to predict the body condition at a finer temporal scale, on a daily scale. Part of the challenge, of course, is that we don't know when the sheep is foraging. Right? That's one of the things about the free roaming animals. So we're trying to infer the foraging behavior from the HMMs to update the covariate for body fat percentage to then include into the HMM again, which is a bit more complicated. But in the general framework, I think there's a key idea of including this physiology into animal movement. And that really should be a bit more, I think, of a priority, because you know your study systems and you know that physiology is going to have a big impact on what they do. So Marie had really nice things to say at the beginning, but I always like to end my talks on the challenges associated with these, right? Because hidden Markov models are great I love all the extensions. I love what I've learned, but they don't come without challenges. One of the biggest things I realized is that the data structure, and we'll talk about two things, sampling efforts for white sharks. So how long you actually collect the positions, how long the tracks are, and label data. The two big issues that I've encountered. So there's a recent paper that came out, this one on ecology evolution. These are white shark tracks for Guadalupe. And they asked me to analyze this movement data. And I was like, sure, but it's 2021. I want to do things in a Bayesian framework. I want to take into account individual variability. I want to do more with my model. Now, these are the 10 sharks that they asked me to analyze. And you might see that some of the sharks have 3 curves and some have 2. Do a little guessing game. Why might some sharks only have 2 densities, the others 3? So we're still trying to capture relatively simple things of short step lengths, higher turning angles, larger step lengths, more directed motions. What we realized is that they collected, for some of the sharks, they collected a lot of data. They followed them for a lot longer. And other sharks have less information. So I quickly realized that for some of the sharks, if I try to say all sharks have 3 states, the ones with the 2 states, I have run into so many computational problems because it doesn't actually have enough information to infer the 3 states for those sharks. If I try to just infer 2 states for all sharks, the 4 sharks above with the 3 curves, it actually didn't fit the data very well. So I'm realizing that trying to account for individual variability and saying all the animals have the same number of states might actually end up being a huge problem when you have a lot of information on some animals where you can actually pick up more types of movement patterns than you can the others. What I did, and you can all criticize and tell me why it's wrong, and I will agree. 10 minutes. I found it very hard, but I thought, well, maybe the 2 states at the end, those extra 2, they're all maybe the same type of sort of traveling or non-error research behavior. And I combined them with these multi-scale HMMs. But I think in practice, when you're collecting data, it's just something maybe to keep in mind. If you're trying to pick up the same number of behaviors across all the animals, the amount of data points you have for each animal might actually have a big impact on what you do. So we have one white shark, and what we try to do here is actually look at errors in search and traveling by depth. So you'd actually see across the time of day when they were traveling at what different depths of the water they were performing this behavior, and errors to search, where were they performing this behavior. So for this shark in particular, it's more at the surface. It is more at the surface during certain hours. And we also did this decoded state counts. So this is Guadalupe, and we have this ARS shallow, ARS mid, ARS deep, and we just overlay that onto a map. Where were these behaviors more evident? And the big red squares are the seal colonies. But I think the challenge, of course, was again the sampling effort, and I didn't realize until recently how much that would affect being able to learn about individual variation. Another big challenge I've had is actually incorporating labeled data. So as I said, HMMs can tell you which observations correspond to which states. But a lot of the criticism as well, do you actually know that that's foraging behavior? Do you see the animal forage? So for the horn sharks, they actually had some label data for the accelerometer data. So for example, this is the horn truck in the line. Great. Oh. Oh. So that one's resting. Yeah, so we have some label data. And so I had a student work on this and said, hey, now we're going to bypass this difficulty of stage ones and really include the label data. And it's going to be better for the things of our states and actually have to validate it. Now, the big thing, the big problem was that there's still a link, even if you have labeled data. I think what's really important is to take into account that you have a behavior in mind, swimming. How does it actually manifest in the movement data you collect? Well, what's counterintuitive is that when we included the labels in the HMM, we ended up with more problems fitting the models, which I didn't think would happen. I thought we include the labels, it should work better. And it didn't. And there's students that are going to work on this, but the idea I think still is, well, is they're just quite varied movements that correspond to the same big behavior. Then maybe it's swimming at an angle, swimming freely, swimming, might be a lot of different types of swimming that we're capturing. And the big thing is that there might be errors in the labels. You might, for accelerometer data that's so fine scale, you might think, well, it's swimming. Then maybe there's a second where it's not swimming. And it's performing a different behavior. So I thought, again, this would be a lot easier. And I discovered a suite of challenges that I never thought about before. Again. And there's a lot of computational challenges, which I won't actually talk about. We don't have too much time. But the idea is that in a Bayesian framework, when you run a Bayesian model, Bayesian hidden Markov model, you get this sort of beautiful conventional output. There's a column on the box that has R hat or ones. We like those distributions, everything looks great. And the problem is, you're usually fitting the wrong hidden Markov model at some point because you're doing this model building. And what that means is that you're likely to end up with finding multiple modes. So try to fit the hidden Markov model. There's these things called starting values. For those of you who have tried it before or will try it one day, you'll probably think back to me today, but you did talk about starting values at some point. And it's a hard problem because you end up with these sort of multimodal distributions. And I showed you when everything goes right, you end up with this pretty behavior. When you fit the wrong hidden Markov model, you might see something like this. So this is just a quick example of fitting a two-state hidden Markov model to data generated from a three-state hidden Markov model. So nothing too complex, but we run into this trouble. But I think the best thing is that when you run into the trouble, is that you have this superpower of having all this biological domain expertise. So you can actually look at the different modes and think, well, if these were my means, how would I explain that in a biological context? If my mean of state 2 was actually 3, what outcome would I think is possible? Right, so looking at all the different modes and translating them into biology is how I've actually learned quite a bit about hidden Markov models and what I actually can get from this. So there's these misspecified HMMs, where you can do simulations. There is a paper that is under revision right now, but it's led by Vianey, and I'm just part of this team where we talk about some of the pitfalls and potential opportunities with hidden Markov models. some of the common challenges that we've come across. So working with continuous time, when your data aren't collected regularly, these hidden Markov models, how could things go wrong? And how might that impact your biological interpretations? And so on. But even though there's a lot of challenges, I always want to leave on a note of hope, is that hidden Markov models are incredibly useful. I got into them because I really wanted to work with shark movement data. But Dr. Marie, here, we were just awarded this grant, Advancing Statistical Methods for the Analysis of Complex Biology Data collected from humans and animals. So it's a team of 15 total. And so we actually formed a team of people that collect sensor data from patients with depression, borderline personality disorder, bipolar disorder. And that same team has people that work on narwhals, Greenland sharks, black sharks, and sheep. Right, so hidden Markov models are not only useful in ecology for animal movement, they're actually useful in ecology for a lot of other things. You can also apply them to a lot of disciplines. And thank you. That is a metal shark that I found in San Francisco that I thought was beautiful. How does the predetermination? Yeah, how does the predetermination of number of states affect the analysis? And is there a way of exploring, analyzing the data to determine how many potential discrete states? Realistically, the number of states depends on a suite of things. It depends on the forms of the state-dependent distributions. It's going to depend on the data you have. So say you found that there's only 2 states that captures the dynamics well, and you obtain more data, you might actually have to increase the number of states. Giancarlo's here from statistics. He's supervised in part by Dr. Carlos Campbell, who's statistics here at UBC, who's across the way. And he actually has this great paper where you might not actually be able to recover the true number of mixture components. There might be a lot of challenges associated with it. So there's also a great paper by Jennifer Poole on practical selection of number of states in movement ecology. So I think they're really the experts, but in part, it just depends on a suite of things. And in part, you also want to find the number of states that explains the data well. So you can look at residuals, you can do simulation-based model assessments, and try to find the hidden Markov model that is representative of a good data generating process for your data, while at the same time taking into account that you don't want a 10-state HMM that you can't biologically interpret. That's not the best question, best answer, but hopefully that's useful. Okay, so the question was, how do you pick the time interval at which you sample to do whatever you need to answer the scientific question? There actually are statistical papers that talk about defining the best temporal resolution for the data. To be honest, I really don't think it's as much of a statistical question as I think it's a biological question of what are you actually, based on the scientific question you want to answer, based on the temporal resolution at which certain behaviors are exhibited, what's feasible. And I think one of the students on Wednesday said they might do something very fine scale and then subsample and understand at what temporal resolution can they no longer identify certain behaviors. So there are statistical ways of doing this, but to be honest, working with ecologists, I just feel like this is really a very biological question. And it also depends on the modeling that you're trying to do. So at one temporal resolution for a hidden Markov model, that might be great. For another temporal resolution for different types of models, you might need a finer scale. So it's probably not a great answer again, but I think internally, at its core, I think it's really a biological question. And you all know your systems way better than any other statistician. So the question was, how do you transition from terrestrial to marine systems in terms of accounting for that third dimension, such as depth? So of course the terrestrial animal is not going to fly. But actually it's related to modeling eagle movements, for example, where they're flying as well. I actually found it really challenging to create states, discrete states that corresponded to things like area restricted search at different depth levels and trying to discretize depth. We did it with the hidden Markov model to try to capture more shallow depths. That's one state, sort of a mid-range, allowing some overlap between what shallow and mid-range behaviors would be. I don't think that actually, I'm not sure discretizing the depths is the best way. And so I'm actually not sure. For the first white shark paper, that we only had GPS data. So we couldn't incorporate depth. And for accelerometers, sometimes we just work with accelerometer data and don't include depth. But I think that's a huge challenge. And I've actually tried to speak with a lot of biologists the thing is, I come in with the fancy models, but you all know your system so well that I think you are really the ones who can say, I want to capture these different behaviors. And this is the way it manifests in the depth data, in the positional data, and so on. So I'm really here to learn from you. So I ran into this, the question was, how do you with sampling effort, deciding how long each time series should be for each animal. Approximately, based on circadian rhythms, do 24 hours. And I think in part, it's going to depend on the study system. So maybe for a sheep that eats regularly every day, it has more predictable patterns, or it's going to exhibit foraging, traveling, and resting most days. You might be able to collect data from at least a day for each sheep and be able to infer what's going on. For other animals that might not have to eat for a week, I think that's going to be a challenge of what do you actually want to capture? Understanding that some days, if you only track the sharks for 24 hours, there might just be a lot of variation across days in what they're doing because they might not actually have to perform the same number of behaviors. So that's something that I'm still, the sampling effort, I can go back and try to answer the question in a statistical way, but then I try to think ecologically what's actually relevant. And again, I think just depends on what you're trying to capture.
So I just want to say first, thank you very much to Dr. Diaz Barajas on the behalf of UBC's Institute for the Ocean and Fishery. And sorry, we're kind of close at the end of time here.